now. So good afternoon, dear students. Today we are going to start our first lecture, that is Introduction to Pharmaceutical Medicinal Chemistry. Okay, so what we are going to actually discuss in this topic. So here we are going to discuss some of the definitions which are actually involved in the study of medicinal chemistry. Those are, so first of all, we will discuss about what is actually pharmaceutical chemistry, or we can call it as a medicinal chemistry. So how it is discovered, how it is developed, what are the stages that are involved in the development of pharmaceutical chemistry or medicinal chemistry, and what are the identification and interpretations of mode of action of which type of compounds, biologically active compounds at their molecular level. So what is actually it is a discovery of something new lead compound, the development of that newly investigated or newly discovered identified lead compound, and their mode of action, how it is acting for the treatment of any disease or any disorder. So this is actually, or these are the processes they are involved in the medicinal chemistry. Basically, it is an interdisciplinary course. Now, the meaning of interdisciplinary means it is not restricted to some of the terminologies, or it is not restricted to the some things. It is interdisciplinary, as it is going to involve the knowledge and terminologies of pharmacology. It involves biochemistry, it involves molecular biology, involves immunology, that is as the drug molecule or the lead compound is going to act on our cell. That's why there is involvement of a life science. So what type of molecules actually involved? So there is involvement of organic compounds, their physicochemical properties, their theoretical chemistry. Now the word theoretical chemistry belongs to how it is acting theoretically. So before actual any lead compound coming to the market, so we study that lead compound thoroughly, the theoretical knowledge, then molecular spectroscopy, what type of structure that drug molecule is going to have, then what type of crystal structure, whether it is amorphous, whether it is crystalline structure, whether it is water soluble, whether it is water insoluble, and some of the chemical properties associated with that molecule, and everything which we are going to combine it in the terms of information technology. So there is involvement of these many terminologies or these many processes with that lead compound, that is a medicinal compound, which we are going to discuss, or so many lead compounds, medicinal compounds we are going to discuss in this subject. Now, before we move actually towards the medicinal chemistry, we know, or we should know, some of the definitions, some of the compounds. Now, we all know we are dealing with, since henceforth, with the terminology, that is drug. Now, in the first year only, we have discussed the definition of drug. What is drug? It is a compound which is used for the treatment and mitigation and prevention of any disease and disorder. Now, in the terms of medicinal chemistry, we are going to define a drug molecule. Basically, it is maybe an organic molecule or inorganic molecule. Now, in the case of pharmaceutical inorganic chemistry, we have discussed so many inorganic compounds which are used. Some are used as hematinic preparations. Some are used as an intra and extracellular acid-base balance compounds. Antacids are there. There are compounds, dental products. So these products are used basically as an inorganic compounds, but they are drug molecules. So organic and inorganic compounds and biomolecules. Now there are compounds coming under the category of biomolecules. Those are proteins and antibodies. Now the example of the protein biomolecule which is used as a drug, that is insulin. There are antibodies, that is penicillin, which is basically an discovery, that is accidental discovery of that drug molecule, but it is naturally exist in the terms of biomolecule. So that activates or inhibit the function of a target cell with the benefit to the patient. So these are the compounds, maybe organic, maybe inorganic, or maybe biomolecules, which will either they are going to activate or inhibit the function of any target cell for the benefit to the patient. So what is the benefit to the patient? He is going to cure or he is going to treat from that respective disease. So these are some of the drug molecules which are used in different type of treatments. That means, that is aspirin, we all know it is used for the treatment as an NSAID. Then sildenafil, it is used for the treatment of, it is used as an male sex hormone. Then another drug molecule, that is paclitaxel, it is used for the treatment of cancer. So these many drug molecules, so the examples of these drug molecules which we are using for the treatment, that is to either activate or inhibit the function of the target cell molecule. Now we are proceeding further for the next definition, that is activity. What happens when any drug molecule either activates or inhibit the function of that target cell? So he is going to show some type of activity. What is that activity? The activity belongs to a target binding place, which is, see, the word is very important, that is stereo electronic compatibility. So the drug molecule, where it is going to act on the target cell for the benefit of the patient, he is going to show the binding type of stereo electronic property with that binding site. Then and then only the drug molecule is going to show the activity. Now the next terminology is low toxicity. Now we know the word toxic, that means it is harmful to the cell. Now the low toxicity belongs to selectivity. So we have to use a drug molecule or select a drug molecule which will be low toxic, which is going to be act on the target cells. So these are the examples of the cells, that is cytochrome, phosphoglycoprotein. So these are the cell on which the drug molecule is going to act. Now the very important terminology is bioavailability. Now what is the meaning of the word bioavailability? That is, it is associated with physicochemical and pharmacological properties of the drug likeliness. Now when we have discussed the terminology of drug, we have discussed that this molecule is going to either activate or inhibit the property of that target cell. Now that drug molecule, this drug molecule has its own some physical and chemical properties. Now how these physico-chemical properties will affect the mechanism of action, the intensity of the treatment, the dose of that drug molecule, that means pharmacological properties, that drug likeliness. Now there are three main phases of drug action. Now in the earlier slide only we have seen the definition of drug, as it is going to either activate or inhibit the site of action of that target cell. We have seen what is the meaning of the word activity, low toxicity. Now how these drug molecule is going to show the activity or going to show the action on these, what type of action? Pharmacological action on that target cell. So now there are three phases of that drug action. First phase is pharmaceutical phase. Second phase is pharmacokinetic phase. And the third phase is pharmacodynamic phase. Now, we all know we are associated with the drug molecules. Those are all pharmaceutical compounds or pharmaceutical molecules which are used for the treatment of any disease. Now, we are not giving that drug molecule as such. We are converting that drug molecule into a formulation. It may be a tablet, capsule, and number of formulations are there. You have seen. So that means we are converting that drug molecule into a dosage form in the form in which we are going to take the drug molecule. So this is the phase or pharmaceutical phase of that drug molecule. That means that drug molecule we are going to take in the form of dosage form. Now, that dosage form, when we are going to take or ingest, will release the drug from that drug form, that is dosage form. Suppose we have taken a tablet of paracetamol. So we are going to take a tablet of paracetamol. When it is going in our gastrointestinal tract, that paracetamol tablet is going to release the drug, that is paracetamol, from that paracetamol tablet. That is, so this is a phase number 1, that is pharmaceutical phase. So that tablet is going to release the paracetamol drug from that dosage form. So this is our first phase. Second phase, pharmacokinetic phase. Now, that is what makes the body with the drug molecule. So step number 1 is over, that is release of the drug from that dosage form in our gastrointestinal tract. This is over. So followed by step number 2 is pharmacokinetic phase. Now, whenever the word pharmacokinetic will come, you are associated with these 4 terminologies, that is ADME belongs to absorption, distribution, metabolism, and excretion. Now, nowadays, one more terminology is added here, that is ADMET, absorption, distribution, metabolism, excretion, and toxicity studies. Now, in the earlier slide only, we have discussed that any drug molecule must be low toxic in nature. So there is one more terminology is added here, but this is for your knowledge purpose only. But we are going to discuss these 4 properties of that released drug molecule. What happens? What our body is going to do with that drug molecule? It is dissolved in our body, absorbed through the blood cells or cell walls. It is distributed in our body. It is metabolized in our body. And after its function is over, that is treatment, the unwanted part is excreted throughout our body. So this is step number 2. Followed by a phase number 3 associated with the drug action is pharmacodynamic phase, associated with what the drug is going to do in the body of human cell or any organism. Pharmacokinetic belongs to what our body is going to do with the drug molecule, and pharmacodynamic belongs to what the drug molecule is going to do in the cell of organism. So it is associated with character and quality of the drug interaction. What type of interaction, what type of physicochemical properties that drug molecule have. So it will going to have some type of interactions with the target cell, with the interaction site, and ultimately on the biological system. So these 3 are the phases of drug action: conversion into dosage form, and in the form of dosage form, we are going to take that drug molecule. So that dosage form will release the drug molecule in the body. What happens after the release of the drug molecule? It is absorbed, it is distributed, it is metabolized, and it is excreted, followed by what the drug molecule is going to do with our body. It is going to have some type of quality of drug interactions with the target cell. So these are the 3 important phases of any drug action when we have taken it for the treatment of either treatment or for diagnosis, anything. Now, before we move actually towards the study of medicinal chemistry, there are some terminologies. What are those? Number 1 will be a target. Already we have seen It is going to interact with any specific cell, that is target cell. So what is that? It is a bio-macro molecule. Micro and macro. Macro means bigger one. So it is a bio-macro molecule to interfere with. So the drug molecule is going to act, going to react, going to bind with a bio-macro molecule, which is known as a target molecule or target cell, we can say. Then this target molecule or the drug molecule will bind with that target cell. How it is going to bind, or where it is going to bind? It is going to bind on the active site of that target molecule. So that active site is known as binding pocket, where it is actually going to bind. See, it is a part of target appropriate to bind a small ligand. So the drug molecule is small in nature, or the structure is too much small. But that drug molecule is going to bind on the appropriate site of that target molecule. That spot or that pocket is known as binding pocket, and that site or that location is known as active site. Now, for example, you know the lock and key. We are not inserting the key everywhere in the lock. We are going to enter the key in that pocket which is provided by that lock. It is true to you. So that key pocket is the pocket given to binding for that key molecule or the key which we are going to use to open the lock. It may be door lock. So that pocket which is provided by that lock used to open the lock by that key molecule. So similarly, all the receptor or the cells have their appropriate locations for individual drug molecules. That drug molecule is going to bind on that binding pocket. So that site is known as active site. Now, next one, a pharmacophore. Next terminology is pharmacophore. What is pharmacophore? It is a part of molecule which is recognized at a receptor site and which is responsible for the molecule's biological activity. Now we know the drug is going to provide us the treatment. It is going to bind on the specific location of the cell. But after all these done, the molecule which is recognized, that cell is going to recognize that molecule. See, the lock is going to recognize the key. Then and then only he will allow the key to enter inside the lock. Similarly, our cells are going to recognize that molecule at a receptor site, and they are responsible for the molecule's biological activity. When it is going to recognize that molecule, that key is going to look like my compounds or my keys, then and then only that site is going to bind on that active site. So this recognition of the receptor site and to show this is responsible to show the molecule's biological activity. Now the next terminology is ligand. What is ligand? It is an organic molecule possessing target affinity. So there is something affinity between the target cell and the drug molecule. So this ligand is going to have some type of affinity, some type of interaction, some type of bonding between the drug molecule and the target site. So that has to be very important, stereoelectronically compatible with the binding pocket. We know the drug molecule is going to bind with the bonding pocket. It is going to be recognized by that receptor cell. Then the next one, it is going to have some type of affinity. How it is going to be bind? So they must have some type of attraction, some type of repulsive forces. Then and then only they are going to bind with that target cell. And these type of forces will involve stereoelectronically compatibility with the binding pocket. Now, what is the meaning of the term active? Active is a compound detected by HTS. HTS is nothing but high throughput screening. Now, when we are going to discuss any drug molecule, We are not directly catching the drug molecules directly from the tree of apple, like we are catching the mangoes from the mango tree, we are catching apple from the apple tree. This is not the process of discovering or identifying any drug molecule. We are going to search, we are going to recognize any drug molecule, active drug molecule, which is detected by high throughput screening. By discovering thousands of compounds, we are going to discover a single drug molecule from that thousand or crores of the compounds. So this is high throughput screening, that is selection of the active drug molecule from high throughput screening. Then HIT. HIT is an active compound which is identified in the screen with confirmed structure and activity that need to be developed into the lead compound. See, in first slide only we have seen the structure of aspirin molecule. See, so the use of aspirin molecule is, it is used as an NSAID for painkiller, it is used to treat the headache and all. Now, so this is a ring structure of aspirin, or this is the structure of aspirin molecule. That means this type of structure is going to show us a analgesic type of activity. So what is heat? Heat is an active compound identified in the screen. Now we know the structure of aspirin molecule. So this is ring structure which is associated with some type of branch structure. So these type of similar molecules we are going to develop on the screen or the monitor or the computer. We are going to confirm their structure and their activities with the cell. Then we need to be developed into the lead compound. Now we know the structure of aspirin. That type of similar compounds we can generate on the screen of the computer. This is nothing but the generation of heat molecule. Now, lead is an active compound with convenient properties. Now we know drug molecules have, it must have target site, it must have binding pocket, it must have active site, it must be a pharmacophore which is recognized by the receptor. It is going to be associated with some type of affinities. The molecule which is going to have all these properties is known as lead compound, which is basically an active compound with all these convenient properties, that is drug likeliness, it is soluble in the water or our gastric mucosa, and it is must be a synthetic feasibility. Easily we can synthesize in the lab. So these all compounds and patentability. No one has discovered this earlier. So this is lead belongs to is an active compound with which is associated with all these properties, that is drug likeliness, solubility, and easy to synthesize, as well as no one has discovered earlier, that is patentability. Now, next definition is drug candidate. So we have seen the active site, we have seen the heat compounds, we have seen the lead compound, and the next one, drug candidate. High activity, good selectivity, in vivo efficiency. From the lead, we are going to discover a drug that is going to have high activity, good selectivity. He is going to have all these properties, as well as in vivo efficiency. See, in the case of heat molecule, we have generated the data on the screen of the computer. But here, in the case of drug molecule, we have to show the in vivo efficiency, means in the body efficiency of that drug molecule. Now, the next definition, that is drug after success in the clinical trials, which is approved by the regulatory agencies, that is Food and Drug Administration and EMEA. So these are all terminologies or the basic terms involved in the pharmaceutical chemistry. Now,